Hi, I'm Heriberto,
Javier.
I'm Adrian.
and we're going to talk about enzymes. Enzymes are proteins that help speed up the chemical reactions. And on an enzyme, inside there's an active site, and this is the enzyme. You can see the blob, and then it helps break down molecules like hydrogen peroxide. And then this is the substrate, and the substrate goes into the active site and helps them break down. It fits in like a key. It breaks down the substrate into different molecules. It breaks down, for example, the hydrogen peroxide into oxygen, one oxygen, and two water molecules. It helps speed up the chemical reactions. And this is the formula.
Alright, so how does an enzyme know when to break down and when not to? Well, that's when you have activation and inhibition, and I'm going to talk about activation. So in activation, you have the enzyme and the active site. But let's say that the substrate doesn't fit in with the shape of the active site. So in order to activate the cell, in order for the substrate to be able to fit into the active site, you would need a coenzyme, which pretty much goes into the active site. And as you can see it's the shape of the substrate. So once it comes inside to this, then that substrate will be able to fit in here. And before the coenzyme binds to the enzyme, you have an apoenzyme, which is pretty much like a premature enzyme. And once the coenzyme binds with the enzyme and it's complete, then it's called the holoenzyme. And another way you can activate the enzyme, another way for activation to occur would be with a cofactor. Now a cofactor is different from this because nothing is going in here. What's happening is a cofactor enters the allosteric site over here, and what it does, it changes the shape of the whole enzyme. So as you can see over here, the enzyme, nothing goes in, but the shape of the enzyme changed to fit the substrate. So now the substrate can come into the enzyme, into the active site. And once again, before it changes shape, it's called the apoenzyme, and after it binds with the cofactor, then it becomes the holoenzyme. And the difference between these two is that a cofactor is inorganic and a coenzyme is organic.
So just like there are ways to activate an enzyme, you can also stop it from activating, and that's going to be called inhibition. There are two ways to stop an enzyme. That's competitive and allosteric. For competitive, you have a substrate and inhibitor with the same shape, and they pretty much compete for further place in the active site. In this case, the inhibitor will go in instead of the substrate, preventing the enzyme from activating. In allosteric, we have the inhibition that goes in through the allosteric site and blocks off the active site, preventing the substrate from going in. The type of allosteric inhibition is when the inhibitor goes in through the allosteric site, changing the shape of the active site, preventing the substrate from binding with the enzyme.
Now when you have when you're separating all those molecules, to measure how much you have, to measure your reaction rate, just to measure exactly how fast the reaction is happening, you would either measure the reactants, or you can also measure the products.